Thank you so much. I think maybe before I start sharing my slide, just a comment about ovarian cancer in general, just to remind us that ovarian cancer actually presents with a lot of symptoms. I know that many times we call this a silent disease, but one of the landmark studies by the group from Seattle several years ago actually show that patients with ovarian cancer can have symptoms. One of the symptoms is abdominal pain, pelvic pain, which occurs in actually maybe up to 70% of patients. So when you have persistent abdominal pain lasting for a few weeks, it is very important to seek medical opinion. So I just want to preface with that before I get into discussing immunotherapy for ovarian cancer. So I'm sure many in the audience will recognize that ovarian cancer have now responded very well to immunotherapy. And so these are a list of questions that I think many people will have. Why has immunotherapy fallen short of expectations, but demonstrated more benefit in many other types of cancers? Are there differences in tumor biology that contribute to this success, especially of immune checkpoint inhibitors that have been approved in multiple cancer types, but not in ovarian cancer? Do standard regimens impact the efficacy of immune therapy, such as radiation or chemotherapy? What are the best biomarkers of response? Are there emerging pathways that can be targeted for future clinical trials? What are some of the ongoing efforts to transform what we now call cold tumors into hot tumors? So I'm going to be, this is a very extensive list of questions. I'm going to be hopefully touching on many of them. I may not be able to get into details, but hopefully give you a flavor for some of the where we are and where potentially we can go. So here's a study published in 2021 that I just want to share, which asks the simple question: does the addition of avelumab, which is an immune checkpoint inhibitor, to liposomal doxorubicin improve the efficacy in patients with platinum-resistant ovarian cancer? This is Javelin 200 study, more than 500 patients. As you can see, the progression-free survival was 3.7 months when you add avelumab to Doxil versus 3.5 months with Doxil alone, 1.9 months with avelumab alone. And again, if you look at the overall survival, no significant differences. So this is one of the initial indications that immunotherapy using immune checkpoint inhibitors such as PD-1 or PD-L1 blockers may not necessarily be efficient. Well, you can argue that those who are heavily pretreated patients, platinum-resistant patients, who maybe their immune system is not quite competent enough. So why don't we move this to frontline? And here is another large study, almost 1,000 patients, Javelin 100 study, asking the question: does the addition of anti-PD-L1 antibody avelumab to standard platinum-based chemotherapy, followed by avelumab maintenance versus chemo alone, does this improve the efficacy? And again, look at the lines, the Kaplan-Meier survival curves. Really no difference. Median progression-free survival, 16.8 months, even when you include maintenance, 18 months with avelumab in combination, and control, again, really no difference. And so this tells us that there is a problem. And then the next study, which is really also very interesting, is what if you add anti-PD-L1 antibody, this time atezolizumab, to standard platinum-based chemo, and now you are adding bevacizumab to increase the efficacy in newly diagnosed stage 3 or 4 ovarian cancer. Again, frontline therapies. Look at the curves. They run very closely together. 19.5 months versus 18.4 months for progression-free survival. And overall survival, no difference. So what are the barriers? Why is immunotherapy not working in ovarian cancer patients? Some of the points that I'm going to make here are actually applicable not just for ovarian cancer, but they seem to be more magnified in ovarian cancer. So you can imagine if you use an immunotherapy, vaccination, immune checkpoints, or even cell therapies, first you need to generate large numbers of immune cells, T cells, that are then supposed to travel to the tumor site. In this case, ovarian tumors occur in the peritoneal cavity. They come to the peritoneal cavity and they encounter multiple immune suppressive barriers. So this is a hostile environment for these T cells to traffic and survive. So upon arrival, they encounter a number of factors. Some of them are cells. There are cells called regulatory T cells, myeloid cells, or bad macrophages. They are called tumor-associated macrophages. There are other chemicals. And then even when those T cells survive in the tumor microenvironment, there's an adaptive counter-regulation, adaptive response. The tumor is fighting back, making additional molecules such as IDO, PD-L1. The tumor is reducing the expression of target antigen on the surface of the tumor. And this leads to a secondary wave of immune suppression, and the T cells become less functional and they are not able to persist. So this is the general model that we and others have studied in detail, showing that these are some of the mechanisms. But look at how complicated these mechanisms are, whereby if you were to go after each of them, you probably will need about 10 or 12 or more drugs in order to counteract many of these mechanisms. So the gaps in ovarian cancer, number one, how do we overcome this complex inhibitory network? Number two, how do we promote effective trafficking of these T cells into the tumor? And number three, how do we promote persistence of the T cells in tumors? So let's start with immune checkpoint inhibitors. This has made tremendous impact. Multiple cancer types, lung, kidney cancers, bladder cancers, name it, except ovarian, and I think pancreatic and maybe prostate cancers. So if you consider a T cell, it has a number of these inhibitory receptors that put a brake on the ability of the T cell to expand and to function. This is a normal physiologic mechanism. All of our T cells, healthy individuals, have this mechanism so we don't have excessive, uncontrolled proliferation and function of our T cells, for example, when we encounter a viral or bacterial infection. So these are brakes on the immune system in order to allow them to stop when they need to stop functioning. On the other side, there are accelerators. These are activating receptors also on the T cell. So there has to be a balance. And we have focused a lot on blocking these inhibitory receptors, such as PD-1, which is the key example. I will come back and talk about some of these other inhibitory receptors because there's a lot of work going on to develop additional antagonist antibodies to block many of these other inhibitory receptors, as well as agonist antibodies to further activate these receptors. Let me show you just one experiment, which I think is very key for us to understand some of why ovarian cancers may not be responding very well to immune checkpoint inhibitors. And I promise this is the only mouse experiment, I think, in my slides, and all the rest are human studies. So So we did this experiment a few years ago where we took mice that were deficient in those inhibitory receptors. Okay, you can see here PD-1, TIM3, CTLA4, LAG3, and so on. These are receptors that are supposed to stop excessive function and proliferation of the immune system. So these mice were knocked out for all of those receptors. And then when you use PD-1 knockout, and then you ask the question in this PD-1 knockout to say, so here's an example, just the PD-1 knockout mouse, and you challenge the mice with ovarian cancer, and then you harvest the T cells from the ovarian tumor-bearing mice, and you ask the question, what is going on with these other inhibitory receptors? So here's a PD-1 where the T cell lacks PD-1, and now you ask the question, how does the T cell express other inhibitory receptors? Look at how high these T cells are now expressing some of the other inhibitory receptors, suggesting that when you block one of these inhibitory receptors, such as PD-1, you can have compensatory upregulation of the others, just like I showed in the previous slide, whereby you block PD-1, these other receptors go up and they limit T cell function. So let me skip this and move on to some of the additional clinical trials. And we showed in those experiments that if you now combine PD-1 and CTLA4, one of those, two of those, you can get better results. But look at this clinical trial. This is GY003. This is a trial led by one of our colleagues, Dimitri Zamarin from MSK, where he treated ovarian cancer patients with anti-PD-1 nivolumab, as well as nivolumab and EP. And so this is anti-PD-1 plus anti-CTLA4. You can see very improved response rates from 12% to 34%. But look at what happened to the progression-free survival. Almost no difference, 2 versus 3.9 months. So this is not a clinically meaningful significant. And then the question becomes, how about biomarkers? Looking for biomarkers in these studies. These are studies looking potentially at the role of biomarkers. And again, at the present time, PD-L1 expression, which has been found to be very useful in a number of other studies. For example, patients with non-small cell lung cancer was found to be not useful in ovarian cancer. So we have a situation where patients are poorly responsive to immune checkpoint inhibitors, number one. Number two, we appear not to have a good biomarker, but I'll come back to that in a minute as well. So then the question is, how about patients with BRCA mutations? Could that be a good biomarker? Again, this is analysis from the paper by that combines chemo with avelumab along with chemo alone. And in this study, again, BRCA mutations, you can see that those patients, if you stratify by BRCA mutation, again, there was no difference. So the mutation was not associated with benefit from chemo immunotherapy combination. How about tumor mutational burden? This was a small study by, again, I think this came from MSK, showing that tumor mutational burden was not associated with improved responses to immunotherapy, as well as HRD. HRD status also not associated with improved responses, although this was predicted to be potentially be able to be associated with improved responses. But I will touch on this later towards the end of the talk. So far, the only signature that appears to have been able to be associated with improved response to a combination of PARP and anti-PD-1, PD-L1 combination is this signature reported from the group of Dana-Farber in 2020, called signature 3. Essentially, it's a signature that is associated with tumors that have that we called inflamed signature. Again, I'm going to touch on that later on. So there appears to be potentially a type of signature of ovarian tumors in a subset of ovarian cancer patients that potentially could predict responsiveness to PARP inhibition plus immune checkpoint inhibition. But this remains to be validated in multiple larger studies. So this is the signature 3 that I talked about from that paper, where patients with signature 3 along the red line in the Kaplan-Meier curve that you can see at the bottom of this slide showed a better response to combination of niraparib and pembrolizumab. So here are just a few additional results. How about if you add bevacizumab? I think we already talked about that. The addition of bevacizumab with or without olaparib in the initial study showed no difference, but there appears to now be some evidence that there may be a signal of improvement in both progression-free and overall survival. This is this Mediola basket trial, which suggests that immune checkpoint inhibitors can provide some long-term therapeutic benefits in some patients. And then we're also going to be talking about TIL, adoptive cell therapy in a moment. But I just wanted to point out also that in terms of immunotherapy, immune checkpoint inhibitors is a major strategy. Another strategy is to take patients' T cells from the tumors, expand them, and give it back to a patient. There's an initial study, very small study, suggesting that there could be benefit where response rate in that patient population was more than 80%. Very small study, so take this with a pinch of salt, including almost 20% of patients who are complete responders. How about CAR T cells? So some of the CAR T cell studies that are going on right now are those targeting folate receptor alpha, as well as those targeting mesothelin and another target called Claudin-6. So those trials are currently ongoing using, targeting folate receptor alpha by CAR T cells, as well as especially in the study with Claudin-6, patients are vaccinated first with an RNA vaccine, followed by the CAR T cells. And in that trial, that is, again, a very small trial, about half of the patients had some evidence of response. So the biomarkers of response are really very limited, as I've shown you. So BRCA mutation status does not appear to be a good biomarker for response. PD-L1 expression, HRD status, all of these do not appear to be appropriate, at least so far, good biomarkers of response if you take a broad population of patients. But from other studies, the tumor mutational burden is one that has been approved for the use of immune checkpoint inhibitors across multiple tumor types. So TMB is now, so patients with ovarian cancer who have high tumor mutational burden could be eligible to be treated with immunotherapy. Patients who have high intratumoral, what are called tertiary lymphoid structures. These are structures that actually are able these are structures that contain a lot of B cells and other types of immune cells, and these appear to be highly predictive of ovarian cancer of immunotherapy in ovarian cancer. So finally, let me just talk about what I think is probably going to come out as one of the best ways for us to immunophenotype ovarian cancer and try and select therapy. So it is now clear that there are at least 3 types. So far, all of our clinical trials, we enroll all types of patients with high-grade serous ovarian cancers, even some studies including clear cell and mucinous types. But in fact, if you look at those tumors under the microscope, there are at least 3 distinct immunophenotypes. One group that is now referred to as the inflamed group. This is the group where you have a lot of immune cell populations, especially T cells, kind of within the tumor microenvironment. So you have T cells, you have antigen-presenting cells, you have B cells, and you have dendritic cells. This is an inflamed tumor. These tumors are likely to react, to be responsive to immunotherapy. Then look at these 2 other categories. One category called immune excluded, where the T cells, they are trying to penetrate the tumor, but look at where they are. They are at the edges of the tumor. So those cells are not able to get in, therefore they are not able to do their job. Okay, so the first category, inflamed, where these T cells are there, they are within the tumor, they have other cell populations, they are likely to respond to immunotherapy with immune checkpoint inhibitors. Immune excluded tumors are unlikely to respond, as well as this last category called immune desert, where, as you can see here, there are really almost 0 T cells present in the tumor. So you need other strategies, especially for the immune excluded and immune desert. We need to come up with additional strategies. So consequently, the strategy that I'm going to spend the rest of the time talking about is a strategy. Actually, I think I need to do this. The strategy that I'm going to be talking about next is a strategy that can overcome the challenges of immune excluded and immune desert. This approach involves the use of oncolytic viruses. These viruses are very unique and special viruses because they've been engineered to attack tumor cells and destroy tumor cells. And in the process of doing this, they release antigens. These are targets that can be recognized by the immune system. And because they are foreign viruses, they are also attracting different immune cell populations into the tumor. So now you can imagine that the antigens, the molecules released from these tumors that are being destroyed by oncolytic viruses can be loaded onto the immune system to activate T cells and generate a tumor-specific adaptive immunity. So this approach actually led to approval of one form of oncolytic virus for melanoma patients. This was way back in 2027. It's called TVEC type of virus. So the question is, if you give oncolytic virus, can you actually leverage this approach to deliver additional compounds into the tumor microenvironment, so that almost like your antibody drug conjugate, which attacks the tumor cells, but at the same time is releasing a drug. So here, oncolytic virus, can we engineer an oncolytic virus and use it also as a delivery system to further modify the tumor microenvironment? And this is, so in order to do that, we said, okay, what kind of additional molecules do we need to engineer and put on the oncolytic virus? So we came up with targeting CXCL12 based on some data, primary data from us by one of my colleagues, Dr. Zeros, who showed in a large panel of ovarian cancer patients that CXCL12 is expressed almost uniformly across multiple ovarian cancer patients. You can see here on the right, this showing that CXCL12 is highly expressed in these patients. So CXCL12 binds to a receptor called CXCR4. So this binding of CXCL12 in ovarian cancer leads to an aggressive phenotype. It leads to metastatic spread. It promotes angiogenesis. It attracts bad immune cells in the population of immune cells that come to the tumor. So the question is, can we block this axis, block CXCR4, CXCL12, and incorporate the oncolytic virus? In this case, we're using an oncolytic vaccinia virus to block this interaction so that we can deliver this to the tumor microenvironment and reverse the immune suppressive microenvironment. So working with one of my colleagues, collaborators, Dr. Cosbo, she initially came up with this concept to arm oncolytic vaccinia virus to block the CXCR4, CXCR12 axis using this compound that I show on the left. I will not get into details of the properties of the compound, but let me just show you very quickly what this compound does when you arm oncolytic vaccinia virus with this CXCR4, CXCR12 antagonist. So here's a mouse model of ovarian cancer. You can see if you don't treat, this is ascites. These are multiple tumor implants. If you use the virus alone, this is what you get. If you use the virus armed with the CXCR4 antagonist, you see significant reduction and improvement in survival for these tumor-bearing animals. And then if you then begin to look at some of the mechanisms, remember what I showed in the beginning, that there are multiple mechanisms within the tumor that tend to suppress the ability of T cells to function. If you begin to look at many of them, such as, again, the role of CXCL12, angiogenesis mediated by VEGF, some of those bad immune cells, myeloid, they are called myeloid-derived suppressor cells, you can see significant reduction. Regulatory T cells, significant reduction. And importantly, okay, this is a model where we did not even vaccinate the animals, but there is induction of immune response against some of the targets on the tumor. In this case, this is WT1. So you can see that CXCL12 blockade of that axis can address those multiple mechanisms of immune suppression within the tumor microenvironment. And then we go on to characterize the nature of the T cells. You can see that there's increased T cells in the presence of oncolytic virus treated with the CXCR4, CXCR12 antagonist, as shown here. These T cells accumulate significantly when you look at the RNA gene expression as well as cells within the tumor microenvironment. And then we also wanted to ask whether, if you recall, we're addressing multiple components. The initial slide that I showed, we want to address trafficking. Can we promote trafficking? Can we suppress multiple mechanisms of can we overcome multiple mechanisms of immune suppression? So those are the things that we're trying to do here. So trying to suppress, trying to improve trafficking. So when a T cell gets into the tumor, it's trying to travel through this through the blood vessels within the tumor, and that involves multiple steps. These are called capture, rolling. There's a step of activation. It sticks to the blood vessel wall. This is the vascular endothelium. And finally, it comes out in order to penetrate the tumor. The question is, can we affect how this is going on? Because if the T cell does not end up in the tumor, no matter how many T cells you're able to generate, it's not going to work. So first, we model this in a mouse model where we ask the question, if we give T cells in this mice, this is normal lumentum, okay, look at the blood vessels. And then if you then inject tumor, you lose vascular integrity. These become abnormal vessels. And then if you then give T cells, these are labeled T cells, again in this mouse model, here's a normal lumentum. This is not a non-tumor bearing mouse. You can see the T cells traveling, they are rolling, all the steps that I described, they are rolling, they are adhering to the blood vessel wall. But if you look at in contrast, this is an ovarian tumor bearing mouse. This is like a graveyard, exactly the same experiment. You cannot see the T cells moving within the tumor. So this tells us that not only should we be paying attention to just giving immunotherapy, we should be asking questions about, are we able to affect the vascular, for example, the vascular diameter? Are those vessels functional? Are the T cells able to roll? Are they able to stick and then extravasate and get into and be able to penetrate the tumor? So I will skip this slide. It's really repeating the same thing. This is quantitative measurement of all of those parameters, showing that in the presence of oncolytic virus, where we block CXCR4, CXCL12 interaction, we can actually improve many of these components. Look at, for example, the rolling T cells and the sticking T cells. They are significantly improved when we use this component. I will skip this as well. So we then asked the question, can we look at this in patients? I mean, I showed you the mouse model, but can we actually look at it in patients? Actually, we see almost exactly the same thing. This is blood vessel in normal skin. This is tumor adjacent to mentum. This is from an ovarian cancer patient at the time of surgery, where we are able to do what is called intravital microscopy. Look at the blood vessels. They are abnormal, they are disordered. And then, and what is so striking is the fact that these blood vessels, many of them are actually non-functional. More than 50% are non-functional. So if you generate large numbers of T cells, whether by immune checkpoint inhibitors, anti-PD-1, or by any of those other strategies, cell therapies, you are unlikely to have efficacy if you cannot affect the functionality of the blood vessels. So all of this has led us now to develop a clinical trial where we will ask a number of questions. This is a phase 1/2 clinical trial. In the phase 1, we have phase 1A and 1B. We ask the following questions. Is the addition of our oncolytic vaccinia virus that is armed to block CXCR4, CXCL12? If you remember, this affects multiple aspects of immune suppression within the ovarian tumor microenvironment. Number one, it promotes T cell trafficking. The virus itself causes tumor destruction of tumor cells. So if we take this and add to standard of care liposomal doxorubicin in patients with recurrent ovarian cancer, is this safe? So safety, phase 1A. Then the next step is to ask the question, if this is safe, if we then add pembrolizumab to that combination, is it still safe? And that is phase 1B. And then if we demonstrate safety of 1A and 1B, then we go to a small phase 2B clinical trial, standard of care liposomal doxorubicin plus OVV oncolytic vaccinia virus that is armed with the CXCR4 antagonist. And cohort 3 is these patients, the liposomal doxorubicin plus the oncolytic vaccinia virus along with immune checkpoint inhibitor. We were supposed to start a year ago. I leave this here because we've been trying to get IND for this clinical trial, and there's one more element that we still need to address with the FDA that we are currently working on. So the primary endpoint is going to be, these are platinum-resistant ovarian cancer patients. Primary endpoint is going to be 6-month PFS rate. I will not get into the statistics. So effectiveness of immunotherapies in ovarian cancer appear to be limited by multiple mechanisms of immune suppression. And effective immunotherapy will require at least a two-step approach, where number one, we reprogram the environment, especially patients with immune excluded and immune desert, where we reprogram that environment with an armed oncolytic vaccinia virus, armed oncolytic virotherapy, armed with, in this case, CXCR4, CXCL12 is what we are targeting. But you can imagine that we can target other pathways using this approach. And the beauty of this is that we actually can deliver directly into the peritoneal cavity of where the tumor really exists. So the goal is to treat intraperitoneally. So number one, reprogram the tumor microenvironment. And number two, generate effective anti-tumor immune responses, either with vaccines or adoptive T-cell therapies. I will not be getting into details. I'm happy to answer questions about this. We have ongoing novel immunoengineering approaches to create T-cells that will arrive at the tumor site and yet be able to resist some of those mechanisms by which the tumor counterattack, and not only resist, but also persist for a longer period of time, so it can be more effective. So these are the approaches that are coming up by our group and some other groups. And at this point, I'd just like to acknowledge our group, my lab members, as well as clinical investigators that participated in some of the our ongoing clinical trials, Dr. Lele, Dr. Zero Satrozo Park, and my co-clinical investigators at the University of Chicago. So let me stop here and take any questions.